In case you want to register with us for any course in natural sciences, mathematics, biology, physics, chemistry, you can get to register with us. We are also offering tuitions in nursing sciences, health sciences, pharmacy, biomed, radiotherapy, physiotherapy, and such. Also medicine. That's just it. You register with us and you get to prevail properly together with us. The ester bond in a lipid formed between the ester bond in a lipid is formed between. Now an ester bond, a lipid has got glycerol, this one. What's the structure? What's the formula for glycerol? Glycerol is C3H8O3. Please differentiate it from trioses, C3H6O3. Trioses, carbohydrates have got a ratio of carbon to hydrogen to oxygen of 1 to 2 to 1. So you can see that glycerol is reacting with fatty acids. In this case, it is a 3 fatty acids, so this is going to be a triglyceride. If you only had 2 fatty acids, diglyceride, 1 monoglyceride. And to be specific, it is only a hydrogen coming from the glycerol is reacting with the hydroxyl group coming from the fatty acid. So option 1 is saying hydroxyl group of glycerol and the hydrogen atom found in the carboxylic group of fatty acid. You can see this is opposite, right? Hydroxyl group is not coming from glycerol, but it's coming from the fatty acid. And then the hydrogen is not coming from the carbon atom found in carboxylic acid, but is coming from glycerol. So that option is not correct. And then option 2 is saying 2 hydroxyl groups, and you can see the functional group is not between 2 hydroxyl groups, it's between, okay, so 2 hydroxyl groups of glycerol, not of glycerol. It is between the 2 hydroxyl groups, but not of, both are not from glycerol. One is from the fatty acid, one from glycerol. And then 3 saying 2 hydrocarbon chains of fatty acids. That's also not correct. And an oxygen of glycerol and hydroxyl group of fatty acid. So that is actually correct. An hydroxyl of glycerol, this one, and so an oxygen actually of glycerol. In this glycerol where you have got OH, you have got an oxygen there coming from the glycerol and the hydroxyl group of fatty acid. As you can see, this fatty acid is going to take that hydroxyl group. Okay, so that's why the option is gonna be this one. 5 is a methyl group of fatty acid and a hydroxyl group of glycerol. That's not true. Which of the following statements is not a characteristic of homologous chromosome? First thing that you need to know about homologous chromosome is these are chromosomes. Like, look at these 2 chromosomes. I'm going to write this one and we can have another one and this one. So what is the first thing you can identify? Homologous chromosomes have got the same length. That is number 1. And then number 2, the same gene is going to be on the same position. Let's say this position, if we're talking about the gene for eye color, even in this position, we have this same gene for eye color. And those different forms of a gene, we call them alleles. The location on a chromosome, we call that as the loci. Okay. And then you can see also this point where the 2 sister chromatids attach, which we call the centromere, is at the same position. So number 1, which of the following is not? They have the same length. That is true, right? They have the same centromere position. That's true. They carry genes that control any given trait at the same location. This is also true. Any given trait. For example, at this point you can have the gene for eye color. Even here the gene for eye color. Number 4 is saying they always have the same type of allele on the same loci. Now that's not true. An allele is an alternative form of a gene. Let's say, for example, from this chromosome, from your mother, it is telling you need to have brown eye color. The other one, the same position, Talking about eye color can be saying you should have blue eye color. So you can see it's not always that the alleles are going to have the same uh, you are going to have the same type of allele on the same position. You can have different alleles but the same gene. The gene is eye color. The alleles blue or brown. Okay, they can be different. So that's why this is not correct. They pair up during meiosis 1 years. Pairing up, homologous chromosomes will pair up during the prophase 1 of meiosis, and that's when genetic recombination or crossing over happens on this position, which we call the chiasmata. Number 3, saying which of the following is correct? Positively charged part of atom move rapidly at nearly the speed of light. The positively charged of the atom is proton. Do they move rapidly at nearly the speed of light? Not really. Okay. The positively charged parts of atoms are the smallest. Are the protons the smallest? No. The smallest protons and neutrons are found in the nucleus. Those are big. That's why they make the atom heavy. The smallest are electrons found in shells. Negatively charged parts of atoms are heavy. This is the opposite, right? Negative are electrons. Electrons aren't heavy. The negatively charged part of atom travel in orbitals. This is true, isn't it? Electrons are found in orbitals, and actually we say what is an orbital. An orbital is a space around the nucleus of an atom where the probability of finding an electron is maximum. The neutral parts of atoms are the smallest. Again, neutrons are the neutral parts, and protons, which are positive parts, these are found in the nucleus, so they actually have the same mass. Number 4, enzymes and other proteins are synthesized in. When you look at the cell cycle, here is it. Cell cycle is divided into two phases. You have got what we call the M phase, and then you have got what we call interphase. In the M phase, that's where you have got specifically meiosis and mitosis. Okay, M phase. And you have got prophase there, you have got metaphase there, you have got anaphase, and you have got telophase. Now, when it comes to the interphase, it is preparation before M phase, which is the cell division. It's going to be divided further. Okay, it's going to be divided like this. You have got the G1, the S phase, and the G2. Can you see that G1 is actually the longest phase? So here we are going in this direction. So a cell will first go into G1. In G1, what we're trying to do is to make proteins and enzymes, because you are preparing that for division. When you divide a cell, let's say you started with one cell and then you have two at the end, the same amount of information that was in one cell should be in another. But for that to happen is you should make sure that you've divided the cell. After G1, actually, a cell can go into what we call G0. G0 is the resting phase. S phase, the prominent thing that happens here is DNA replication. In G2, what will be happening is that anything that wasn't made in G1 will be made. That is growth too. For example, we'll be talking about the duplication of organelles. They are going to start growing. Organelles will start duplicating in G2. So enzymes and other proteins, it's in G1. Identify the expected phenotypic ratio of a dihybrid test cross between heterozygous dominant phenotype. So heterozygous dominant and recessive homozygous phenotype. Remember, this is a dihybrid. So in a dihybrid, you are talking about T traits. So let's say, for example, we're comparing someone is tall, and let's say you're not just talking about being tall, you're also comparing, say, eye color. So let's say, for example, capital T is being tall, and then capital B is having blue eyes, and then small t is being short, small b is going to be being having brown eyes. So this is a dihybrid. So in these traits, what you see is, before you start crossing for the Mendelian genetics, you are going to start first with all is the homozygous dominant and homozygous recessive. So we're going to have capital T, capital T, capital B, capital B. You are going to cross this with capital T, or sorry, small t, small t, small b, small b. So for the Mendelian genetics, you always start first with the dominant trait cross with a recessive trait. When you are crossing these, the first products that you are going to see, we call that as F1 generation. You are going to have a combination, everything, all the products will be a combination of capital T, small t from here, and then capital B, small b. This will be the combination for everything. And then to form F2 generation, what you want to do is now for you to be able to cross each one of these individual F1s. And how do you perform that cross? That cross, first you get this T, pair up with that T, so that will be capital T, capital B. Get the T again, pair up with that, capital T, small b, and then pair this small t pair up with this one, so that will be small t, capital B, and then this pair with that, small t, small b. Now start crossing. If these come together, what are you going to form? If these come together, what are you going to form? Just like that. That is how you go about this one. Now you are going to put all this on the same, on every point, small t, capital B. So let me explain now using the question so that you get it. So we've been told that the expected phenotypic ratio of a dihybrid H cross is we have got heterozygous dominant. Heterozygous means, let's say someone has got a capital T, they also have a small t. This is for one trait. Remember, this is a dihybrid, so this is for height, it is heterozygous. This is how we're talking about eye color also. It is also heterozygous. And you are crossing with a recessive homozygous. Recessive means small t, small t, small b, small b. So before you can cross these ones, you need to make sure that you make alleles as they are going to be found in gametes. So first this T will cross with the B. That is what gives you capital T, capital B. And then the same capital T will cross with small b, capital T, small b. This is the one. And then get the small b, the small t, cross it with the capital B. That will give you this one. And then the same small t crosses now with a small t. That will give you this one. So write it on this row and also on this row, and then start crossing them. For here you can see we are going to have capital T from here, small t from there, capital B, small b. You are going to do for all of these which are here. And we are trying to look at what? You are looking at the phenotypic ratio. Phenotype are the physical traits. So look at those that have got at least a capital T in them. At least, I've said at least, meaning that even one T, which is capital, and then they should have at least a capital B. It doesn't matter whether there are two capital B's as long as there is even one. And then you look at those where you have got a small t, only you don't have any capital T, but you have got a capital B. And then you look at those where I've got a capital T, it doesn't matter whether it's a small t with another small t or capital T, but you should always have a small b. And then you can look at that with a small t and a small b. So this one which is having a capital T and capital B, this one is going to be tall with blue eyes. The second one is going to be short with blue eyes. The third one is going to be tall with brown eyes. The last one is going to be short with brown eyes. So if you can see, there is a capital T here and a capital B, capital T and the capital B, capital T and capital B, even this one. So these are four. So we'll say four. The total that we are dealing with here, if you count these 1, 2, 3, 4, 5, 6, those are 16. So this is 4 out of 16. If you go to the one where you have got only capital small t's and then a capital B, at least one capital B, you have got this, 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 this. This is also 4 out of 16. And then we have got a capital T with a small b, capital T, at least one capital T, small b, even this, even this, even this. This is also 4 out of 16. And then we will just have smalls here, 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 here. That is also 4 out of 16. I just wrote that on the wrong position. So you can see the ratio is 4 out of 16, 4 out of 16. We can say this is 4 to 4 to 4 to 4. And if you write this as a ratio, that will be 1 to 1 to 1 to 1. So this is what you need to know about a cross between a recessive phenotype and a heterozygous dominant. Study the organic molecule below and then answer the questions. Identify the non-reducing sugar. Now, because I can see that here we have reacted two sugars, I can tell these are disaccharides. And out of all the disaccharides, it's only sucrose, which is a non-reducing sugar. Why? Because in sucrose, all the functional groups have reacted. And you know, sucrose is made up of glucose plus fructose. And one special thing you need to know about fructose is that it is the only hexose sugar that is a furanose. Furanose means it looks like a pentagon, like this. So it's only this structure there. Okay, it's the only one. So this is carbon number 2 of fructose. This is carbon number 1 of glucose. Because both function groups have reacted, there is no free function groups that will be able to reduce other substances. That's why sucrose ends up being a non-reducing sugar. So it is 2. The atomic and mass number of a neutral sodium atom is that. So they've given us atomic number being 11. And then they've also given us mass number being 23. This is for a neutral sodium. And then they give say what is the mass number of sodium ion. Now you know for sodium ion nothing has changed. The number of protons, which is the atomic number, is still 11. The number of neutrons also hasn't changed. It is still the same number. How do you find neutrons? Neutrons, you get the atomic number removed from the mass number. So that is a 12. It is still the same, meaning that mass number also hasn't changed. It is only the number of electrons that have changed. So the mass number, the question is saying, what is the mass number? The mass number is supposed to be 23. So if you look at these options, there's no 23 there. So that is just a mistake. Okay. Number 8 is saying, dash can induce changes in electrical charges of neighboring molecules. Hydrophobic interactions, ionic bonds, covalent bonds, Van der Waals interactions, hydrogen bonds. Van der Waals interactions, what happens that when you have got two atoms like this, now for the atom we have got negative charges, right? So when you bring to get close negative atoms, what will happen is that the electrons will start to repel each other. And so what you are going to see is, let me say I get this one. You see electrons are going to be pushed and they'll concentrate on one side. The side where they've concentrated will become partially negative because of the electrons. The side where they've moved will become partially positive. So Van der Waals forces can induce changes in electrical charge in neighboring molecules. Okay. Next question is saying, which of the following molecules represents unsaturated fatty acids with one double bond? So unsaturated fatty acids. First, a fatty acid looks like this. And then you have got a double bond with oxygen, and then you have got OH. So you can see this fatty acid doesn't have any double bond, meaning that this is a saturated fatty acid. You don't consider the double bond with oxygen, that is part of the function group. This is a saturated uh, this is a saturated fatty acid. If you look at the formula for this guy, at the end here, you have got a carbon bonded to two oxygens and an hydrogen, right? Okay, but let me start from this end. You have got a carbon is actually going to have three hydrogens, and then you have got this carbon with two hydrogens, this carbon with two hydrogens, this carbon with two hydrogens. So I can simply say CH, and then I can put N there. N can be any number of carbons with two hydrogens. Actually, CH2. Remember, each carbon has got two hydrogens. And then you have got the carbon that contains the functional group COOH. So this is the functional group for saturated fatty acids. But if they become unsaturated, you are going to have HO. our bond with oxygen, we are going to have something like this. Okay, so what changes in this case? What has changed is that at this point where we're supposed to have, of course, we're supposed to, let me just put another one there so that we maintain the same. So at this point, initially, we're supposed to have two hydrogens, isn't it? But because of the double bond, we only have one hydrogen because each carbon can only be bonded to four things. Same with this carbon, we only have one hydrogen because each carbon is supposed to be bonded to three things. So the formula that we have, when you have got a monosaturated fatty acid, you are going to remove a 2 from it. So the formula becomes CH3, CH2, and then you are going to have N minus 2. We have removed 2 from the number of hydrogens, okay, and then COOH. So this will become, now I need to be specific, not the way I've written it, because it's specifically only the night on the hydrogens. The formula that I wrote was including the carbon also. So I'll distribute first this N to carbon. So I'm going to have Cn and then H2N. But that 2N is from where I'm going to move it to. It's just from the hydrogen that you're moving it to. Okay, so this is the formula. So another way in which I can write this is, can you see that you have got a carbon here, a carbon here, how many carbons? Two carbons. And then I've got three hydrogens plus this hydrogen. How many are they? Four hydrogens. So this is the ratio of 2 to 4, which is the same as 1 to 2. So basically it is the same as what I wrote here. Carbon to hydrogen is 1 to 2. So meaning I can, without writing this and without including this carbon and this hydrogen, I can still say we are supposed to have CH2, okay, 2N. This is the initial what you're supposed to have. But because we have got a double bond, we're subtracting a 2 from that. We are remaining with two oxygens. So this formula and this formula is actually the same. So CnH2n minus 2 and then O2. Okay. I just forgot to distribute this nitrogen. Remember, we're distributing it here also, so it's going to be here. So that's the formula for monosaturated fatty acid. Next question is saying dash substitution is a type of gene mutation where a dash is replaced by a dash. So again, if you just look down here, you have got adenine and guanine. Adenine and guanine, we call these as purines. And then we have got cytosine and thymine. These are called as pyrimidines. So where you get one pyrimidine and substitute it for another pyrimidine, or you get one adenine and substitute it for another adenine. So for example, you add A, C, C, G. After mutation happens, you'll now have A, T, C, G. Can you see that where there was T, C, we are now having T? That's a mutation. But because cytosine, which was there, has become thymine and both of them are pyrimidines, we call this as a transition. But if you change one pyrimidine for a purine or one purine for a pyrimidine, we call that as transversion. So transverse is purine for pyrimidine. This is correct, isn't it? Transition is purine for pyrimidine. That's not correct. Transition is thymine for guanine. You can see that is still thymine. Is, you can see that's a pyrimidine. Guanine is a purine. So that's still opposite. That's still transversion. And then transverse is pyrimidine. Pyrimidine, that's also not correct, right? Okay. Transition is cytosine positive uracil. Is that correct? Cytosine uracil. Now uracil is also here. Uracil is also a pyrimidine. So what is this one saying? Transitional cytosine positive uracil. Okay, so whatever the positive there is saying, that is not correct. You know, for though you have got the cytosine, the pyrimidines being cytosine, uracil, and thymine. A substitution here, if we get a cytosine for and replace it for uracil, that can still be considered a transition. But the cytosine positive uracil, we don't know what that statement means there, the positive. In other words, let me just say that 5 would have been a potential answer. But because you have got something that is more specific, a substitution is the one that you are getting, isn't it? So now let's look at the question again so that we don't make any mistake. Actually, this is transitional. Okay, transitional, yes, so that will supposed to be correct. So let's say we have dash substitution is a type of gene mutation where a dash is replaced by a dash. So transverse substitution is a type of gene mutation where a purine is replaced by a pyrimidine. That's transverse. Which of the following is the strongest in aqueous medium? So which bond is the strongest? Of all the bonds, you need to know that covalent bonds are the strongest, followed by ionic bonds, followed by hydrogen bonds, and then followed by van der Waals forces. So covalent bonds are the strongest in aqueous medium in water. Bacteria such as E. coli grown on nitrogen 15 medium and transferred to nitrogen 14 medium and allowed to grow for one generation. DNA extracted from these bacteria is centrifuged. What density distribution of DNA would you expect? Remember that first it was grown in nitrogen 15. Okay, meaning that all the DNA for this bacteria has been marked with nitrogen 15, and then it is now transferred in nitrogen 14 and allowed to grow for one generation. So what is the density of distribution? So here what you do is, let's say the blue color represents nitrogen 15. So this is the DNA that we have grown, right, in an environment containing nitrogen 15. So this is an experiment which was done by Nelson and Stowe to prove Michelson and Stowe to prove that DNA undergoes semi-conservative replication. So what happens that when you have transferred it now from a nitrogen 15 medium to a nitrogen 14 medium, if it grows for the first time, what happens that this strand will split. They split like this. So because they are now in a nitrogen 15 medium, nitrogen 14 medium, sorry. So let's say red will be nitrogen 14. What happens is each strand is going to get a strand that is marked with nitrogen 14. This will be the first generation G1. Even this strand will get like that. If we allowed this to replicate again, what you see is that each one of the strands is again going to separate. So I'm going to have two strands which are blue, right? And then you have got two strands which are red. But because the environment is still the environment of nitrogen 14, this will be a second generation. So each of these strands will gain another red strand which is having nitrogen 14. And nitrogen 14 we call it light nitrogen. So what can you see from here? In the first generation, all the all the offsprings are hybrids. Hybrids means they have got equal amount of nitrogen 15 and nitrogen 14. So these are hybrids. If you look at the second generation, you have got these two which are hybrid. So you have got 50% hybrid, and then these two are the new strands containing nitrogen 14. So I've got 50% new strand. Okay. Another way we can say this is we can use another ratio. How many blue strands can you see out of all these? So I've got 2, 4, 6, 8. Out of all the 8, you only have 2 which are blue. So you would have also said 2 out of 8. That is 25%. So 25% heavy nitrogen, that's nitrogen 15. And then 6 out of the 8 is actually light nitrogen, it's in red. So 75% nitrogen 14, which is light. So this is another way in which we could have represented the same information. So because this was just after first generation, we are interested in this. So only high density, is this only high density? No. One high density and one hybrid, is that true? No. Or only high, so it's supposed to be only. Okay, so only hybrid nitrogen 14 and 15. This is true, right? This is only hybrid. And then half low density and half hybrid. That's supposed to be second generation, isn't it? Only low density. That's also not correct. Next, a group of students crossed F1P plants that had round and yellow seed in a dihybrid cross. The results of the F2 generation were as follows: 85% round yellow seed, 26% round green, 28% yellow ring seed, wrinkled yellow, and 9% wrinkled green. Determine the chi-square value of these. So how you do this is F1 generation, okay, that had round and yellow. Now this is a dihybrid cross. For a dihybrid cross, when you are having traits, the F1 generation, you're supposed to cross. Let's say we're talking about the round being capital R, and then if it's not round, the other trait is what? Wrinkled. That the wrinkled is going to be small r. And then let's say yellow is capital Y, the other color is green. Let's say green will be small y. Okay, so this is what we have in first. Before you can have a first offspring, remember I said that for the hybrid cross you always start with capital 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 capital, and then you cross with this small 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 small. So the F1 generation, all the offsprings when you cross will have a capital and a small, okay, and then a capital Y and a small y. So because we've been told that the F1 had was round and yellow, the only thing is because the offsprings that are born after the first generation in a Mendelian monohybrid cross, the offspring that are born in the F1 generation are dominant traits. So meaning that capital the dominant trait here, the dominant trait is for R, round is dominant over wrinkled, and then for the color, yellow is dominant over green. Now for it to get a second generation, it's supposed to cross this with itself. And what you need to know is that for a dihybrid cross, when you cross like this, you get a genotypic ratio, a phenotypic ratio of 9 to 3 to 3 to 1. Okay, this is what you need to know. You can perform it. I already showed how you can perform a cross. First pair this capital R with this small, with this capital Y, so that will be R Y. Pair again this capital R with a small capital R small y, and then get the small r pair up with this, that is capital, and then get a small r pair up with this to be small small. You write it there, also write the same here, and then you start crossing. Like for example, you're going to have capital 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 capital. You do for all of them. You'll see that those which are going to have a capital R and at least one capital R and one capital Y, they are going to be 9 out of the total. Those that are going to have a capital R and a small y and only small y's will be 3. Those which are going to have only small r's and at least one capital Y will be 3 also. And then those which are going to have everything being small, just be 1. So that is 1. This is the normal ratio, Mendelian ratio for a dihybrid. Now that we know this Mendelian ratio, we can write the traits and then be able to calculate the chi-square. So if you just know the ratio, there's no need for you to be able to cross. If you know the ratio for the hybrid cross. So the first one, the first feature, we are going to have those that are yellow and round. So round and yellow. Then we are going to have those that are, if it's not round, then it's C. So the first feature is round and yellow. So we're going to be having observed results. I can put all there, the values that we have seen from here. And then what are the values that we are expecting Those are expected results from the Mendelian ratio. So round and yellow, how many are they? They are 85. That's the observed. And then the next is round and green. And those are 26. And then we have got wrinkled and yellow. Those are 28. And then we have got wrinkled and green. Those are 9. So if we find the total of the observed results, we have got 85 plus 26 plus 28 plus 9. So the total is 148. But remember we said that the genotypic ratio, the expected genotypic ratio from the Mendelian genetics, this is supposed to be 9 out of 16, because the hybrid we find a total of 16. It's supposed to be 3 out of 16. It's supposed to be 3 out of 16. This is supposed to be 1 out of 16. So what you do is get the total after you add these. That's 148. So to find the expected round yellow, you say 9 out of 16 times 148. So I'll say 9 times 148 divided by 16. So the expected value here is 83.25. And then again, we are going to go, we'll say 3 out of 16, which is here. And then we multiply by the total, which is 148. That is going to be a 27.75. That will be the same here, 27.75. The expected for wrinkled and green is 1 out of 16. And then you multiply that by 148. So that is supposed to be a 9.25. Actually, when you add also this, they will give you 148. So from the chi-square, the formula that we use for the chi-square, okay, so the chi-square given as x squared is given by the sum of the observed values minus expected values squared divided by the expected values. Okay, so we'll start. Chi-squared is going to be, first, get a first to subtract them. So get the observed is 85, subtract the expected, which is 83.25. The answer that you get, square it. So it has given me 3.0625. Remember, supposed to divide that by the expected. The expected is 8, 3.25. Plus, go to the next. You are getting 26. You are subtracting 27.75. You square it. The answer that I've gotten there is 3.0625. Divide by the expected, that is 27.275. And then plus, go to the next. Subtract 28 minus 27.75. The answer you get, to square it. That is going to be a 0.0625. And then you divide by the expected. The expected was the same, 27.75. And then you do the last one, which is 9 minus 9.25. You square the answer. That is going to give 0.0625. Divide that by the expected, which is 9.25. So if you add up these values, you are going to get 0.156. So that is the calculated chi-square. So the next question is saying, using the chi-square table, choose the appropriate conclusion. So now what you do is compare the calculated chi-square to the tabulated chi-square. Okay, so remember this is a dihybrid cross. A dihybrid cross has got how many characteristics or how many physical traits? There are four, isn't it? Round, green, wrinkled, and also yellow. So these are four different traits. So we say n is equal to 4 for different traits. You can find what we call the degrees of freedom. Degrees of freedom is given by n minus 1, so that will be 3. Now for chi-square, in physics, in chemistry, in biology, we work at 95% confidence. This is equal to 0.05. Now this is a value, like the calculated, the tabulated chi-square for a monohybrid and hybrid. You can just have them on your brain, but you can find them from the chi-square table. So we're talking about 3 degrees of freedom and at 95% confidence, which is 0.05. You can see that is 7.8. So this is something you can know, because in some questions they're going to ask you to determine what is the tabulated chi-square for a dihybrid. A dihybrid is always 7.8. For a monohybrid only has got 2 traits. So the degrees of freedom will be 2 minus 1, which is a 1. So monohybrid, the tabulated value is always 3.8. So we are using 7.8 there then. So we are saying the value that we have calculated, we said, is 0.156. That's calculated 0.156. The one on the table is 7.8. So can you see that the one we have calculated is more than the one that is on the table? Every time what we have calculated is small than what is on the table, it means that there is no significant difference. Okay, there is no significant difference. In other words, the reason as to why we didn't get the actual values that are given, like the actual expected values, would be maybe because of some normal errors, some expected errors. So the option 2 there, the observed values are not significantly different from the expected values. If what you have calculated is more than what's on the table, there is a significant difference, and so you are going to reject your findings. 15 is saying, what role do hydrogen bonds play in the properties of water? Now hydrogen bonds are the ones that increases the specific capacity. They are the ones that makes water to have a high melting point. This is what makes water to have a high heat of vaporization. In other words, the properties of water is because of hydrogen bonds. They decrease the boiling point. No, they actually increase the boiling point. They contribute to the high specific heat capacity. That's true. They make water molecules less cohesive. They actually make water molecules to be more cohesive. Okay, like the attraction between each water molecule be stronger. They reduce the melting point of ice. They actually increase. They increase the adhesion between water molecules. Now, water molecules are not held by adhesion, it's by cohesion. So that's not correct. Number 16, choose the statement which is true about alleles. I already mentioned this, isn't it? Alleles are alternative forms of a gene. For example, let's say, for example, you have got capital T, small t. These are all talking about height, but this is for being tall, this is being short. They are both alleles, but they are talking about different how. So a gene answers what? What trait are you talking about? An allele answers which one? Are you talking about being tall or being short for that kind of gene for height? Okay. So the next one is saying genes are the alternative form of allele. This is the opposite. It's allele is the alternative form of a gene. Different alleles of a gene are located on the same chromosome. Now, different alleles of a gene are located, they are going to be located on different chromosomes, one from your father, one from your mother. Traits are alternative forms of an allele, and that's also not correct. Actually, traits in Mendelian genetics refers to these same genes. Alleles of linked genes are located on different chromosomes. Alleles of linked genes are located on different chromosomes. So linked genes, these are genes that are close to each other. So for example, I've got this. So I've got, let's say this gene is connected to another gene. Let's say this is talking about blue eyes. This is talking about having brown hair. If these are linked genes, they are close to each other. They are found on the same chromosome. So even in being inherited, they'll be inherited together. That's why you see most of the time there are some traits that you always see together. For example, someone has blue eyes, maybe they're going to have brown hair, something like that. For example, okay. So linked genes are found on the same chromosome. Number 17, hydrogen bond will form between hydrogen and any atom that contains oxygen, fluorine, and nitrogen. So they can form in water molecules, a nitrogen atom in a molecule and a hydrogen atom in another. This is also true. Hydroxyl group, that's the OH, on carbon 2 of glucose residue that forms amylose. This is also true. Okay, amylose polymer. NH and carbon to oxygen group in the polypeptide. So the NH bond, you have got hydrogen there. And then for the COO, you see, you still have, so this can still form hydrogen bonds. So this is all of the above. Which of the following bonds is the weakest? We already mentioned this. We said covalent are the strongest, followed by ionic, followed by hydrogen bonds, and then van der Waals forces. Flagellin is a projection on the cell surface. Now, if you have got bacteria have got flagella, right? The protein that makes up flagella is flagellin. So a projection on the cell surface is flagella, not flagellin. Flagellin is a protein that is part of the flagella. An organelle in prokaryotic cells is not an organelle. A protein found in the cell wall, that's not again a protein of the cell wall. A glycoprotein found in projection on the cell surface, that's not a glycoprotein. A protein found in projection of the cell surface, that's true. It's a protein found in the projection is the flagella. Next, gene mutation in sickle cell disease is an example of. Now, a gene mutation in sickle cell disease in examples of missense mutation. What happens is that in sickle cell, at position number 6, there is a change. There is actually a change in the arrangement of amino acids, and then it causes the change of glutamate into valine. Okay, so a missense mutation is a type of mutation where, let's say for example, you had ACC. When a mutation happens, let's say you become ACG. Let's say this codon was forming an amino acid A1. After a mutation, you're still forming the same amino acid A1. This mutation is known as a nonsense mutation or a silent mutation. But let's say it undergoes a mutation and then you form AGC. But this codon doesn't form the same amino acid. Instead, you are forming amino acid B1. So because you are ending up with a different amino acid, we call that as a missense mutation. Okay. And that's what happens in sickle cell. How does the electron, it's supposed to be electron configuration of an atom affects its biochemical activity? Remember, electron configuration tells us of the electrons that are in the atom shell. So it determines the atom's weight. Weight is determined by protons and neutrons, not electrons. It changes the atom's nucleus. It doesn't change any nucleus because the electrons are found in the shells. Determines how atoms interact with each other. Yes, chemical reactions are determined by the electrons. Stabilizes the inner electron shells. Not really. Prevents the atom from emitting radiation. Not really. Identify the type of side chain in this amino acid. Now you know that amino acids, they've got the general formula like this. Actually, this hydrogen sometimes you can see bonded to this side, so we're going to have Ha3N carbon COO minus. This will become positive. Then you have got a hydrogen. You have got the arrow group. We call this as the deuterium nature of the amino acid. Now, the arrow group is what determines the properties. If you have got arrow group being hydrogen or carbon to hydrogen bonds, a lot of them, these are non-polar bonds. So if you see such an arrow group, that means that you have got a non-polar compound. And actually, the amino acid where the arrow group is an hydrogen is glycine, the same place amino acid. So this is a non-polar guy. This is what determines the acidic properties of an amino acid. So if you had nitrogen here, you would tell that that amino acid is going to be acidic. If you had COOO, you would tell that that's going to be basic. Why are lipids such as oils partially soluble in water? Now, lipids like oils, let's for example, I just talk about a phospholipid. You have got the tail, which is non-polar, and then you have got the head, which is polar, right? So this can interact with the head. So the head, which is polar, can interact with water, and then the tail that is non-polar will be able to repel water. But here, this is any lipid. So they form hydrogen bonds? No. If they form hydrogen bonds, they will be fully partially. They will be fully insoluble. They carry no charge to interact with water. Necessarily not, because if there's a partial interaction, it means that there should be some charge. They have a non-polar portion that repels water. That's true. They dissolve but do not form a homogeneous mixture. They don't actually dissolve, they only interact. Only a certain portion will be able to interact with the water. They are denser than water, that's false, of course. Any molecule which acts directly on an enzyme to lower its catalytic rate is called, what do we call such a molecule? That's an inhibitor. And some inhibitors are going to bind, let's say this is the active site. This site we call it the allosteric site. Any other site, inhibitor that attaches to the active site, we call that as a, this is going to be, what's the term again? Okay, how come it's going just now? Okay, such an inhibitor that attaches to this site is known as competitive, and then the non-competitive attaches on the allosteric site. Imagine forgetting competitive. And then the next question here, study the picture below and answer question 25. You can see that. Which of the following properties is beneficial to living organisms in the picture? What makes insects be able to walk on water is because of surface tension. Surface tension is because of hydrogen bonds created by water molecules on the surface of water. So these water molecules are held by cohesive forces. Okay. So surface tension is due to cohesion. An oxygen atom can react with two hydrogen atoms to form a water molecule. Which type of bond is formed in this reaction? And we've been told an oxygen to form in water. You know, in water you have got a covalent bond, but this covalent bond is polar because there is an unequal sharing of electrons. Why? Because oxygen loves electrons, so electrons are much closer to the nucleus of oxygen. So that's polar covalent bond. Which of the following fatty acids will have the highest melting point? C18 stands for carbon atoms, while 0 to 4 stands for double bonds. What you need to know is that for compounds that don't have, this is the fatty acids, this guy here is saturated. If you have something that is unsaturated, this is monosaturated, it has one double bond. If we had two double bonds, it disaturated, tetrasaturated. But for these guys that are saturated, they are normally going to be solids. For the ones that are unsaturated, they become liquids. And compare between a liquid and the solution is easier to melt. It's a liquid, isn't it? So which of the following acids will have the highest? So highest melting point, the one that is difficult to melt, is a solid. So it is the one that doesn't contain any double bond. followed by the one with one double bond, followed by the one with two, followed by the one with three. And then this one will be the easiest to melt because it will be very, very liquid. Why is ice able to float on liquid water? There's only one reason. Because ice is less dense than water. So the density of ice is lower than that of liquid water. Option one is saying water freezes at 4 degrees. So 4 degrees is actually the water is densest at 4 degrees. If you increase the temperature and if you lower the temperature, the density drops. You can see this is anomalous. Okay. At 4 degrees, that's where this is going to be density. Density is 4 degrees. At 4 degrees, you have got highest density. And then beyond that, it's dropping, and less than that, it's dropping. So water is actually ice at 0 degrees. The density of ice is higher than that of liquid water, no. Density of ice is equal to that of liquid water, no. The mass of water molecule in ice is higher than that in liquid water. That's also a no. The ratio of hydrogen atoms to oxygen atom in galactose. Now, monosaccharides obey the formula CN, H2N, or N. Can you see that? Hydrogen to oxygen is 2 to 1. So they obey that formula and they obey that ratio. What is the function of the sigma factor in transcription? You know, transcription is a conversion of DNA into RNA. Now, DNA is first a double stranded molecule. You need to separate this before you can convert it to RNA. Now, transcription is different from replication in that replication involves a lot of enzymes which perform different functions. For example, in replication, what you'll be separating this strand is going to be known as, let me tell you a bit about replication. The first thing that will first remove supercoils so that a molecule can become like this. It has removed supercoils. It's known as topoisomerase, also known as DNA gyrase. And then what will come and start removing this double bond so that now what will come and start removing, disconnecting the phosphodiester bond, not the phosphodiester bonds, but the hydrogen bonds between the guanine and cytosine and adenine and thymine will be now DNA. In this case, if it is replication, it's helicase. And then you are going to have a protein attaching this side to prevent this from coming together because that has a single stranded binding protein. And then what will be coming to make a copy of the DNA is known as the DNA polymerase. But for transcription, it is RNA polymerase that is doing everything, separating the strand, making a new strand, and you don't have single stranded binding protein. That's why this side, after you've separated it, to still come together. But for RNA polymerase to locate the position of the DNA template where you need to start transcription, you need what is called the sigma factor. It guides RNA polymerase to the correct site of the DNA template. Number 31, the nucleosome A surrounds nuclear pores as a DNA and non-histone protein. It's fully responsible for packing DNA into chromosome, has only RNA, contains a core histone with RNA wrapped around it. So you can see how this chromosome is formed. You first have this strand of DNA having the phosphodiester bonds and the hydrogen bonds. Now, when you start wrapping DNA around histones, these are histones, that's when you form a nucleosome. So that's why this one which says as DNA and non-histone is not correct. And then surrounds nuclear pores, not necessarily. So is fully responsible for packing DNA into chromosomes. So once you have got these nucleosomes, like the long chain of a nucleosome, it will now start wrapping around itself, packaging, and that forms a chromosome. All right. So contains a cohistone with RNA, it's not RNA. So that is what happens. Post-transcription or modification of premature messenger RNA includes the following except. So when our RNA has been made and we want to modify it after transcription, it has got the 3' end and the 5' end. So what happens that on the 3' end we are going to be adding adenines, a lot of adenines, a lot of them. So I've got adenine, adenine. So we call this as a poly A tail. So addition of poly A tail, and that's at the 3' end. At the 5' end, we add what we call guanosine triphosphate. So I've got guanosine triphosphate cap, 5' guanosine triphosphate cap. But this same guanosine triphosphate, we quickly add the methyl group on it on the 7th position. Okay, on the 7th position, we add the methyl. So this is actually known as 7-methyl guanosine triphosphate cap. So addition of methylated guanosine head, that's also true. And then when you form your mRNA transcript, you are going to have portions that don't take code for proteins, we call those as introns, and then portions that code for proteins, we call those exons. So we'll be removing introns and connecting exons. Connecting them, we call that splicing. So we remove introns, we join exons. So termination of transcription is not post-transcriptional. It is actually happening before transcript, during transcription, because in transcription we're going to have initiation, elongation, and termination. These are three phases. But after transcription, that's when we have these other modifications. 34. The number and sequence of amino acids in a protein is called a DAS structure. So the number of amino acid, let's say we've got amino acid A, bonded to amino acid B, bonded to another one, into another one, another one, another one. We call this as a primary structure. The primary structure can end up folding, and it can fold into something that is going to be spiral, or something that will look like this. So we call this as the alpha helix, this is as the beta pleated shape. And then this shapes what this is the secondary structure. And secondary structure is held by hydrogen bonds. Primary structure is held by peptide bonds. So the secondary structure, which is held by hydrogen bonds, of course, for you to have a secondary structure, it means that you already have primary, so you also have peptide bonds there. Now this one can further undergo even a more greater folding. Some of them will form a protein that is like this. We call this as globular, and majority of the enzymes, their shape is globular. Or you can have a shape that is, long like that. We call that as a fibrous shape. So fibrous and globular, this is a tertiary. If you get a lot of secondary tertiary structures, we put them together. For example, you get this tertiary, this tertiary, this tertiary, this tertiary. That is going to form now what we call a quaternary structure. For example, hemoglobin has got 4 tertiary structures, 2 alpha, 2 beta, to form a quaternary structure. So the arrangement of amino acids, the sequence is the primary structure. Next is saying which functional group are present in glyceraldehyde. Now all carbohydrates, like for example glyceraldehyde, I've got OH, you only have 3 carbons. All carbohydrates have got a carbonyl functional group, and then they've got a hydroxyl functional group. Okay, all of them. Which of the following sites of the lac operon prevents RNA polymerase from transcribing the structure gene in the presence of glucose? So I can talk about the lac operon a bit so that you can just understand what we refer to when you are discussing this lac operon. Okay. So the first thing you need to know about the lac operon is that you have got 2 genes. So this is your structure of DNA. DNA is going to have too many genes. You have got genes that helps in forming proteins that are going to be used in structures, and then the genes that will regulate the formation of those proteins. So we are going to have 3 important structural genes. So these guys, we call them structural genes. And then we have got the regulatory genes. Again, for the regulatory genes, we have got a lot of them. We can start from here. We have that. We have this. So these are regulatory genes, and you need to know what they are. So this one that I've written here is known as like A, like I. This is the promoter. This is the operator. Now remember, when you are converting DNA into RNA in transcription, DNA polymerase attaches to the promoter. So I can put this common structure here. This is DNA polymerase because it needs to transcribe the DNA. But what happens in this case is that to the operator, you have got a gate that is attached there. So you have got this guy here. So what this guy does on the operator, what do we call this guy? This is known as a repressor. So the repressor is going to be preventing the passing of the DNA polymerase to go and transcribe the structural genes. And so every time the repressor is attached on the operator, there's not going to be any replication happening. And specifically, these genes that we have here are helping, you are forming proteins that help in the breakdown of lactose. So the only way you are going to form the proteins that are going to be helping in the breakdown of lactose is you should have no glucose. Completely glucose should be used up. Number two, you should have the presence of lactose. Okay. Once you have got lactose, once lactose comes and attaches on the repressor, this is going to be an instruction. The repressor is going to remove its hands from the operator, and this way is now open, and the DNA polymerase can now pass and transcribe the genes. Okay, so this is what happens. Important to know there. What is the function of lac I? I is inducible. Lac I is present behind the promoter, as you can see, and it's the one that makes the protein repressor, which stops this function of the operator. And once the repressor is attached to the operator, we call that as the bound state. Okay, so that's important for us to know there. And then these structural genes that we can see here, these are three structural genes. We have got this one, which we call lac Z, and then we have got this one that we call lac Y, and then we have got this one that we call lac A. Each one of them have got a function. So starting with lac Z, lac Z will code for the enzyme which is known as a beta galactosidase. Galactosidase. What beta galactosidase does is that it breaks down lactose into glucose and galactose. And then lac Y codes for the enzyme known as beta galactoside permease. Galactoside permease. And what this does is that it's going to make cells to be more permeable, to take up the lactose, so that when you take up that lactose, you can now break it down. And then lac A codes for the enzyme which is known as beta galactoside transacetylase. Okay, so this is what happens. So now that we've discussed which of the following sites of lac operon prevents RNA polymerase from transcribing the structural genes. So remember we said this the operator, isn't it? That's the structure of the lac operon. The lac operon is this entire structure here. It's because of the repressor attached to it. The atomic number is determined by the number of atomic number is the number of protons. Choose the correct statement. Glucose is a structural isomer of galactose. No. Glucose and galactose only differ at carbon number 4 in the way the position of the OH. Only at carbon number 4. So in one molecule the OH is on top, in another one the OH is down. Actually the way glucose is, if you look at its open chain, it's like this. This is OH, this is OH, OH. You're actually going to have another OH, and then you have got CH2OH. This will be hydrogen, this will be hydrogen, this will be hydrogen, this will be hydrogen. So the way you see glucose, the appearance of glucose is that if you follow the OHs, they make an S like that. Position number 4 is this one. Because the position number 4 is on the right, it means that when you form the ring, it will be at the bottom. So position number 4 of glucose, the OH is down, while galactose, it is on top. So glucose and galactose, because they just differ in orientation in space of atoms, we call these as stereoisomers. Fructose is an optical isomer of galactose. It's actually an optical isomer of glucose, not galactose. Ribose is the structural isomer of deoxyribose. Now, fructose is not an optical isomer of glucose also. Sorry for that. They are structural isomers because fructose is a ketose. Glucose and galactose are aldehydes. So fructose is actually a structural isomer of galactose. They have got the same molecular formula. Ribose is a structural isomer of deoxyribose. They are not structural isomers because they are different in number of atoms. Ribose has got an oxygen, deoxyribose doesn't have an oxygen. Galactose is an optical isomer of glucose. Now these are not really optical isomers. Instead, they are stereoisomers. Glyceraldehyde is an optical isomer of dihydroxyacetone. Now these guys also, they only differ at one carbon, so they are actually not really the same so it's not like they differ at one carbon, but they have got the same molecular formula, but they have got a difference in the way the atoms are arranged, in the way atoms are arranged. So these guys are structural isomers, glyceraldehyde and dihydroxyacetone. Okay, so from what we've said, this is telling us that of the options that we have, none of them is even correct here, because galactose and glucose, those guys are supposed to be stereoisomers and not what is here. Those are stereoisomers because they only differ in the arrangement of atoms in space. Hope that makes sense. Optical isomers, unless if you have got one which is D and then one is L. Okay, D glucose and L glucose. But we can take them as optical isomers when we are taking the open chain because they are differing at carbon number 4. But just take note, galactose and glucose are stereoisomers. Choose the correct statement about fatty acids. They are completely hydrophobic. Now fatty acids, I've got this formula. So there is the end that loves water. So they are not completely hydrophobic. They are made up of glycerol linked to long chain hydrocarbons. That's not a fatty acid. That would be now a triglyceride or monoglyceride or diglyceride. It's a glyceride which is made up of glycerol and a long hydrocarbon chain. They are both hydrocarbon chains and carboxylic group. They have both hydrocarbon chain and carboxyl group. That's true. This is the hydrocarbon chain. This is a carboxyl group. They are completely hydrophilic, that's also false. They are amphoteric, that's also false. Amphoteric being both hydrophobic and they are amphoteric in nature. Okay, so the amphoteric, something that is both acidic and basic. It's not true. Actually, amphoteric means hydrophobic and hydrophilic. There is amphipathic and there is amphoteric. So you need to differentiate those two terms. When you look at the way water is, water can interact with both something that is hydrophobic and something that is hydrophilic. So it is amphipathic. Okay. And then amphoteric, yes, amphoteric means both acidic and basic. And this one is not going to be true, right? Identify the incorrect statement about coenzymes. They are small organic molecules that can loosely bind to enzymes. So this is true. Coenzymes, an enzyme and a coenzyme is inactive. So this is true. They transport chemical groups from one enzyme to another. That's also true. To make enzymes active, most vitamins act as coenzymes. Yes, that's true. Okay. So coenzymes activate the enzyme. But coenzymes that are ions, we call them cofactors. They are required to activate enzymes. That's true. They are covalent bound to the enzyme. So covalent bonding is strong bonding. They are not covalently bonded there. Okay? They just interact with the enzymes because covalent bonding means that to be permanent. Which of the following is incorrect about the role of tRNA in the process of translation? Each amino acid has its own tRNA which carries a triplet anticodon. This is correct. Each amino acid has got its own tRNA with a triplet anticodon like this. So the longer end is the 3' end. That's where the amino acids attach. And then you have got CAA here. CAA. This is the anticodon which carries. This one is the anticodon. That's where the tRNA will be able to pair with the mRNA complementary strand. 2 is saying acts as an intermediate molecule between triplet codon of mRNA and amino acid. This is true. Once the tRNA, the mRNA gives you the codons, you use now those codons to tell what amino acid you are going to get, and the tRNA is what gets them. 3' end of tRNA carries a sequence of 5' GGA, so this is not correct. It's CCA. Okay, the anticodon on tRNA is complemented to specific codon. This is also true. If the codon is AUG, the anticodon will be GAC. And then the tRNA is an adapter molecule as it has one end that can read a triplet codon in mRNA. This is true, which is the anticodon. 42. Choose the correct partial electric charge involved in the formation of hydrogen bonds in water. So here is water. Oxygen is partially negative because it's more electronegative. Hydrogen is partially positive. Okay, so it's this one. Which of the following is a rare or is or are rare major functions of lipids in the human body? Lipids will store energy. They are going to be used as structural components in the cell membrane, for example, phospholipid bilayer. They also used to make hormones, for example, the thyroid hormone. They also for insulation, like if you have got a lot of lipid, it acts as an insulator. Also used for buoyancy. Okay, so it's all of the above. Someone who is homozygous has homozygous means two copies which are of the same allele. For example, capital T, capital T. Capital T is an allele, capital T is an allele, or someone that has got small t, small t. These are also alleles. They are homozygous. Two copies of the same allele for a gene. This is a gene for height, but they are the same copies. Choose the ketose sugar out of the following. Glucose is an aldehyde, galactose is an aldehyde, mannose is an aldehyde, glyceraldehyde is an aldehyde. The only one here is fructose, which is the hexo sugar. Point mutation involves, now point mutation involves a single base. We call it the substitution. For example, if you have got ACC, you change it becomes AGC. A mutation has happened. There's a substitution there. Change in a single base. Which one of the following results into an efficient positive regulation of the lac operon? Presence of repressor, presence of an inducer, presence of glucose, absence of lactose. Remember, as we said earlier, on the lac operon, you have got this as your DNA molecule, and we said that we have the structure, three structure genes there, and then we have got regulatory genes, the operator, the promoter, and then we have got lac I. Okay, like that. On the operator, we have got this guy, which is a repressor, which is acting as a gate. So the presence of a repressor inhibits the lacoperon from functioning. The presence of glucose also inhibits. Absence of lactose also inhibits. Presence of both glucose and lactose also inhibit. As long as glucose is there, the lacoperon will not work. Lacoperon only works when you don't have glucose and you have got lactose. Because once you have got no glucose, the body wants to have another way by which it's going to make energy. That's when you break down lactose, right? Now, an inducer, the inducer is the same lactose, presence of the inducer. That's it, lactose. So once you have got lactose binding to the repressor, the hands will be released here, and then the operator is going to be free for RNA DNA polymerase to pass and transcribe the genes, the structural genes. Gene mutation occurs at the time of DNA replication, DNA repair, cell division. So during replication, this is where most of the mutations happen. Okay, during the replication. In the cell cycle, remember, we had the M phase, and then we have got G1, S, G2. It's during the S here where replication is happening. Which of the following is not a function of proteins? Enzyme, that's true. Transport molecules, that's true. Like for example, in the cell membrane, you have got these transporters for ions. Defense, that's true. Enzymes and antibodies. Energy storage. Now, energy storage and less lipids, right? And carbohydrates. Structural support. Some proteins can be, of course, involved in the support of structures. For example, collagen is found in our bodies as part of our connective tissue. The protein tertiary structure is primarily due to interaction between the amino and carboxyl group of amino acids, hydrocarbon chains of amino acids, peptide bonds between amino acids, side chains of amino acids, two or more polypeptide chains. Remember, we said that the tertiary structure, you first have primary, held only by peptide bonds. You have got secondary, held by hydrogen bonds. Now, tertiary, you are folding the structure even all the more. And the reason as why I'm folding the structure is because the side chains can interact. You are going to have hydrogen bonds there. You are going to have hydrophobic bonds. You are going to have ionic bonds. You are going to have what we call disulfide bonds. Okay, so all these bonds you'll be able to see them are going to have what we call hydrophilic bonds. So all these bonds are found in tertiary structure because of the side chains. What are the side chains? The ara groups of an amino acid. Remember, you have got ara, carbon, hydrogen, COO minus. Then you have got NH3. Okay, like that. What are expected positions of hydrophilic and hydrophobic amino acids in a globular protein? So when you have got a globular protein, as I told you, a structure that is like this, because it has folded, and what you expect is that the hydrophobic guy should be hiding inside because you don't want them to interact with water. Water is on the outside. Okay. So the hydrophilic end should be outside. So hydrophobic amino acids will be inside and hydrophilic will be outside. Prokaryotic first use all of the following dash in the prokaryotes first use all the dash in the media to obtain energy. So it's glucose that you are going to use first. Once the glucose is done, you can now start using other forms of energy. What is a stop codon? You know, when translation is happening, you have got the start codon AUG, which is a start to signal the starting of translation. Stop codons is a codon that will be, it doesn't specify any amino acid. Once you see that codon is means stop, you have got UAA, UGA, UAG. These three are stop codons. This is the start codon. Does not specify any amino acid. Codon where transcription ends. The codon where transcription ends. This translation or transcription. The end of a chromosome. No, it's not really the end. The place where synthesis of DNA ends is the synthesis of proteins. The last amino acid which is translated is not an amino acid, right? Which bond is not associated with enzyme substrate interaction? Hydrogen bond, ionic bond, van der Waals covalent hydrophobic bond. Remember we said the enzyme and substrate actually just like the enzyme and the inhibitor. These bonds are normally not covalent bonds because covalent bonds are permanent. Okay, for permanent inhibitors, they can bond by covalent bonds. Same with the substrate. The substrate doesn't bond forever with the enzyme, so it doesn't use covalent bonds because those are strong and they are permanent. Enzymes speed up chemical reaction by what the enzyme does is this. If this is the activation energy barrier, what the enzyme is going to do is going to come and lower this activation energy barrier. Okay, so lowering the activation energy, making exothermic reaction become endothermic, no, and the opposite, no, increasing time required for the substrate to reach the transition state, no. Transition state is the top here. It will actually reduce the time it will take there because once you've reduced this here, to take just a little time for you to reach the top. Increasing the activation energy, you don't increase, instead you lower. Choose the false statement about the structure of eukaryotic chromosomes. Eukaryotic chromosomes, DNA and proteins are in equal proportion. Now, this is not true. This is okay. Choose the false statement. DNA and proteins are in equal proportion. This is going to be true, yes. DNA, is transcribed into RNA and that forms proteins. They're in equal proportion. The histone protein, like when you have got this as your, this is your DNA, it will wrap around an histone protein. And then the same amount, again, you are going to have a histone. Same amount histone. So that's the meaning of DNA and protein are equal in proportion. Histone proteins that are found in chromosome are basic, and that's true. These histones are basic. There are 5 types of basic histones. That's also true. Proteins and DNA are not in equal proportion. So it's just the opposite, isn't it, of what we've talked about. DNA wraps itself around the histone, and this is true. Ribosomes are made up of, a ribosome is actually RNA, specifically rRNA and proteins. This is what makes up a ribosome. So I've got the small ribosomal subunit, the large ribosomal subunit, those coming together with the rRNA to form up a ribosome. During translation, the role of the enzyme peptidyl transferase. Peptidyl transferase is the guy that is connecting. If you have got this as your tRNA, and this tRNA is carrying an amino acid on the 3' end. You have got another tRNA that is also carrying an amino acid. To connect these amino acids by forming a peptide bond, you need peptidyl transferase. So it forms peptide bonds between adjacent amino acids. Building blocks of nucleic acids are, now in nucleic acids we have got nucleotides. What's a nucleotide? You have got a phosphate, you have got a pentose sugar, and a nitrogenous base. Minus a phosphate, we call that as a nucleoside, but nucleic acids, DNA, RNA are made by nucleotides. Identify the most appropriate proportion of the nitrogenous base in DNA according to Chagaf's rule. So according to Chagaf's rule, you know that there is a complementary base pairing. Adenine in DNA pairs with thymine, while guanine pairs up with cytosine. So if you have got 10% of adenine, you're supposed to have 10% of thymine because they're supposed to be in equal proportion. So just look for that one where they are equal proportions of the complementary bases in DNA. So if adenine is 22, thymine should be 22. If guanine is 28, cytosine should be 28. Okay. So that's Chagaf's rule. Which position of a codon is set to Ubo? So the third position is set to Ubo. For example, if I've got UAA, UA, let's say UAG. So you can see these positions UAA, UGA, UAG. The third position can be different, but you are still going to code for the same thing. Like these are stop codons. So a lot of codons, for example, can have CUU, and then you have got CUG. The third position is different, but these two different codons still code for the same amino acids. So we say the amino acid, I mean the third position of the codon wobbles. Number 62, identify a non-membranous structure from the following, which doesn't have a membrane. Now, of all the organelles, it is only a ribosome, at least that is of the common organelles that you know is only a ribosome that doesn't have a membrane. That is why ribosomes are found in prokaryotic cells. Prokaryotic cells are not supposed to have membrane bound organelles, but ribosomes are found in prokaryotic cells because the ribosome is not a membrane bound organelle. The rest, endoplasmic reticulum, Golgi apparatus, nucleus, chloroplast, these are not found in a prokaryotic cells because they have got a membrane. Which of the following describes the function of the rough endoplasmic reticulum? Rough ERA contains proteins, so it contains ribosomes, and ribosomes are used to synthesize proteins. So isolation and transport of proteins to their site of need, synthesis and transport of lipids, not really lipids. Lipids is for the smooth. Breakdown of lipid soluble toxin, no. Destruction of worn out cells, really no. Synthesis and transport of carbohydrate, no. So it's about the synthesis, isolation, and transport of proteins. Once you form these proteins, you can now transport them to the point of need. 64, the enzyme dash removes the supercoils in the DNA helix in front of the replication fork, while dash separates the double-stranded DNA into single strands. As we said earlier, topoisomerase, also known as DNA garase, will be removing the supercoils. Helices will be separating the double-stranded molecule. This is in replication. Which of the following molecules carries genetic information for prints? What carries the genetic information is DNA. And this DNA will transmit that genetic information into mRNA, and then from mRNA to proteins. So step 1, this is known as replicate transcription. Step 2, translation. You also have actually replication where DNA is making a copy of itself. These are three phases: replication, transcription, translation. We call that central dogma of molecular biology. 66. The amino acid attachment arm on transfer RNA is found. I've been showing this for long now. This is your tRNA. The 3' end, which has got CCA actually, is where amino acids are attached. Blocking of an um, enzyme action by blocking its active site is called dash inhibition. So this is competitive inhibition. Non-competitive is where there is the inhibitor binding to the allosteric site. In crossing a homozygous recessive with heterozygous, what is the chance of getting a offspring with homozygous recessive phenotype. So we are crossing homozygous recessive, let's say small r, with heterozygous capital R, small r. So look at this. This will be capital small. This will be small small. Capital small. This will be small small. So what is the chance of getting homozygous recessive? You can see this is half, right? 50%. Which of the following statement is correct about prokaryotic RNA polymerase? Prokaryotic RNA polymerase. It carries out the replication. Now, what carries out replication is DNA polymerase, not RNA polymerase. RNA polymerase is for transcription. It establishes hydrogen bonds between nucleotides in a double-stranded DNA. Really not again. This is RNA polymerase, so it's nothing to do with forming double bonds in DNA. It is involved in DNA transcription. This is true when you're converting DNA to RNA, that is the transcription of DNA. Synthesizes both introns and exons. The synthesis of introns and exons. For prokaryotics, you don't form both introns and exons unless eukaryotics. It combines with proteins to form a ribosome. Now, the prokaryotic RNA won't combine with any protein to form any ribosome. Post-transcriptional capping involves addition of. Now, this is a cap, 5 prime. The cap is formed at the 5 prime end. I already explained earlier when you have formed your RNA, you have got the 3 prime end, you have the 5 prime end. At the 5 prime end, you are adding guanosine triphosphate. The guanosine triphosphate, you quickly add a methyl group at the 7th position, so we call this a 7-methyl guanosine triphosphate cap. So methylated guanine to the 5 prime end, that is what we call capping. The 3 prime end, you add a poly A tail. The mitochondria have dash phospholipid bilayer, while cell membrane have dash phospholipid layers. The mitochondria have dash phospholipid layers, while cell membrane have dash. Okay. So the mitochondria has got 4 phospholipid layers. You know the meaning of that? The cell membrane, the mitochondria has got a phospholipid bilayer. The cell membrane also has got a phospholipid bilayer. But now here, if you look at a bilayer, phospholipid bilayer, you have got this one. Actually, the cell membrane, the mitochondria has got 2 membranes, the outer membrane and the inner membrane. And this is made up of a phospholipid. This is phospholipid, so 2 plus these 2, that's 4. But the cell membrane only has got a phospholipid bilayer, and that's only 2 phospholipid layers. If the sequence of bases on the template strand of DNA is a 3 prime T C C G A T 5 prime, the sequence of bases on the mRNA transcript will be, so this is on DNA. What will be the mRNA transcript? So one thing we need to know is, first, when you have got a DNA strand, you have got one strand running from 5 prime to 3 prime direction. You have got the other strand running from 3 prime to 5 prime direction. The first thing is you need to get this DNA and convert it into what? Into mRNA. But the strand that you use is the strand that is running from 3 prime to 5 prime. Okay? That is the one you use in forming mRNA. And this is the one. And it is now this guy that you are going to convert into your proteins. But here we are looking for the sequence of bases on the mRNA. You now want to form mRNA, because if this is DNA has to be replicated, isn't it? Yes, it is replicated. But okay, since we already know that that is the DNA in the 3 prime to 5 prime, what will be the RNA? RNA will be complementary. 3 prime will pair with 5 prime. T pairs up with U because of forming RNA. C pairs up with G, C with G, G with C. A pairs up with, supposed to pair up with T. Okay. Actually, the first one here, the T is supposed to pair up with what? Thymine is supposed to pair up with adenine, isn't it? And then here, adenine is supposed to pair up with thymine, but you are forming RNA, so we don't have thymine, so we're going to have uracil. And then again, T pairs up with the adenine, and then you are going to have 3 prime N. So this is supposed to be the answer. 5 prime end A, G, G, C, and then you're supposed to have UA. Okay, the sequence of bases on the mRNA. So which one is that? AGGCUA. So this is actually option 5, not option 4. Please correct that. Identify the structure that is absent in prokaryotic cells. We have a cell in prokaryotic cells. We have got a plasma membrane. We have got ribosome. We have got plasmid. Plasmid, these are circular DNAs. But I said every membrane bound organelle is not found in prokaryotic cells. Things like the mitochondria, the Golgi apparatus, the any lysosomes, those are not found. During DNA replication in eukaryotic cells, which enzyme adds RNA primers to the lagging strand? Now I've got two strands. Let me put it like this. So I've got this one strand, and then you have got this other one. This one says the one running from 3 prime to 5 prime. This one is from 5 prime to 3 prime. And this is replication happening. So in replication, DNA polymerase can only read the strand running from 3 prime to 5 prime. So it can only make a new strand in the 5 prime to 3 prime direction. But for this strand, we call this as a leading strand. The first thing that happens is that primase is going to lay down one primer. A primer is a nucleotide. Okay, it will lay down only one. So it will lay down one primer. And then what will happen is that the DNA polymerase will now continue forming other phosphodiester bonds connecting other primers, connecting other nucleotides. But for the strand running from 5 prime to 3 prime, we call this as the lagging strand. What happens is that primase doesn't lay down one primer. Instead, it is going to lay down a lot of primers. A lot of primers. And then what will happen is that DNA polymerase will come and form short connections, short joints amongst these short joints. And those short joints, we call them Okazaki fragments, those short joints. And then the one that will come and now finish forming the phosphodiester bond, the full connections, will be known as DNA ligase. So the question is saying, during DNA replication in eukaryotic, which enzyme specifically adds RNA primers to the lagging strand? It is a primase to be the one which is adding primers on both strands. Which of the following statements is incorrect? New DNA strand is synthesized in the 5 prime to 3 prime direction. Is that true? Yes, this is true, as I've said from the top there. Even for the lagging strand, RNA polymerase will still be working from 3 prime end to 5 prime end, except it will be working in the opposite direction. Here it will be going in that direction. Here I've got another one going in the opposite direction. 2. Prokaryotic DNA has more than one origin of replication. More than one origin of replication. So in prokaryotics, these ones we call them monocitronic. They have got only one origin of replication. It is eukaryotics that are polycitronic. The RNA primer are cut out from the newly synthesized DNA molecule. Is that true? Yes, that is what happens. RNA primers will be cut off from the new strand that you formed because they are RNA and you are forming DNA. Leading DNA temperature is always 3 prime to 5 prime towards the replication fork. That is true. This is known as a replication fork here. Lagging DNA template is always in 5 prime to 3 prime towards replication fork. That's also true. 76. Which of the following statements best describes semi-conservative replication of DNA? The replicated DNA consists of two odd strands. That's false, right? So if you add this as your strand you started with, after replication happens, they will split like this. So can you see that you have got one odd strand and one new strand? Okay, so this is not correct. Each strand is replicated of replicated molecule as a mixture of odd and new DNA segment. Which of the following statement best describes each strand of replicated molecule as a mixture of old and new? This is not always the case because let's say another replication happened. The blue guys will also separate and the red guys will also separate. So I've got four strands like that. And then what is happening is that the new strand that you are going to have, the new strands are blue. So this will be the one. Can you see that this one and this one, they don't have any odd strand, so there's no mixture there. So that statement is not completely correct. The replicated DNA consists of one odd strand and one new strand. Yes, this is true. Okay. This one strand is old, this one new strand is new. Even here, this strand is old, this strand is new, even if they're of the same nature. I already explained this when we were talking about Michelson and Stau's experiment. The replicated DNA consists of two new strands. Not really. Replicated DNA is single-stranded. That's not true. Initiation of DNA replication takes place at we have got what we call the origin of replication. So first what will happen is that DNA protein attaches to the origin of replication and that will tell helicase to come and start separating. Before helicase, you have located the origin of replication here using DNA. And then topoisomerase is going to come and remove the supercoils. And then helicase will come in and now start breaking the hydrogen bonds. Okay. And then DNA polymerase can come and synthesize a new strand. 78. Esterification is a reaction that occurs between dash to form glyceride. Now, esterification, we already explained this earlier. For example, in glycerol, you have got OH of glycerol and it will react with BOH of a fatty acid. So it is hydroxyl group and carboxylic group. So I've got OH and you have got this bond. So this is a carboxyl group. This is your glycerol. I mean your OH hydroxide. Which of the following is correct about DNA synthesis on the lagging strand? Phosphodiester bonds between adjacent nucleotides break. Now you are not breaking bonds, you are forming. Bonds between nitrogenous bases and deoxyribose sugars break. Again, you are not breaking any bond. The leading strand produces Okazaki fragments. That's false, right? It's the lagging. Hydrogen bonds between nucleotides of the two strands break. You are breaking no bonds. The gaps between adjacent DNA fragments are sealed. Yes, sealed by actually ligase. Which of the following properties of water that is or are involved in surface tension? It is cohesion. We already explained earlier on when we are talking about that other question which had an insect. In prokaryotic transcription, which of the following factors most significantly determine the initiation of RNA synthesis? Now, the sigma factor, remember we said that is the one that will direct where RNA polymerase is polymerase 2 actually, where it's going to bind. Yes. So this is the most significant to determine initiation. Termination factor is for stopping. Start codon, really not. This is actually replication. So about the start codon, that is in the translation. The elongation factor, that's elongation also. Release factor, so also termination. An enzyme inhibitor that resembles a substrate is called, this is also competitive because it will bind to the same site where the where the substrate is supposed to bind because they have got the same shape. They resemble each other. The function of lysosome in eukaryotic cell is to synthesize protein, no. Digest micro, yes, these are digestion. That's why lysosomes are found in phagocytic cells. For example, I've got what we call macrophages, which phagocytose. So they're going to have a lot of these lysosomes to digest cells or any, not just cells, but anything that is foreign to the body. Which of the following statements is incorrect? Polyadenylation occurs at the 3' end. This is true. Polyadenylation occurs at the 5' end of tRNA. That's false. Not 5' end, not even tRNA. RNA splicing involves removal of introns. This is true. rRNA contains small RNA molecules. This is also true. What role does glycerol play in the structure of triglycerides? So it's like this. The rest are hydrogens, of course. This is glycerol. And this guy will come and react with the fatty acids. So what role does glycerol play? It is a structure to which a fatty acid molecule attaches. Really, they don't attach, but they react. But we want the raw, not what is happening. It is a backbone to which three fatty acid molecules attach, and that's true, right? This is specifically a triglyceride, so it's three. It is a catalyst in the formation of triglyceride, really not. Provides the carboxyl group, no. It provides attachment for two fatty acids, no. This is a triglyceride, unless if it was diglyceride. Which one of the following statements is correct? All the information in DNA codes for proteins. This is not true. You have got some portion that don't code for any protein, like the introns. All the information in nature, in mature mRNA transcript code for protein. Yes, the mature is because you have undergone modification, so you have removed the introns. Both the strands of DNA and of DNA are transcribed to form mRNA. This is, so when you look at this is transcription. For transcription, when you're forming mRNA, you don't use both strands. You only use the strand that reads from 3' end to 5' end. The other one, you don't use it. So you call this as the non-coding strand or the template strand. The prokaryotic primary transcript undergoes capping, polyanhydration, and splicing. This is correct. Those are modifications. The primary transcript is immature. You need to make it mature by undergoing modification. Prokaryotic primary transcript contains introns and exons. That's also correct there. That's not correct. I had mentioned it earlier on one question that prokaryotics don't contain introns and exons. It's only eukaryotics. So that's why this is false. Okay. Even option 4, the prokaryotic primary transcript undergoes. Now, this is also false. These modifications are for eukaryotic cells. My bad. For eukaryotic cells. So you correct that one. That's also not correct. Both strands of DNA are not transcribed. That's also not correct. During elongation stage of translation, mRNA gets attached to the large subunit of the ribosome. No. So mRNA doesn't get attached to the large subunit. So initially what happens that when mRNA is formed, it will locate the small subunit, and the small subunit will start moving along the mRNA. Once it reaches to where you have got a start codon AUG, this will signal the coming of the large subunit to come and attach. This is in initiation. Okay. And then now you read the AUG and then you command a tRNA to bring the amino acid methionine, which is the first amino acid. tRNA binds to the small subunit of the ribosome. It doesn't bind to the small subunit to simply come in and bind within this complex compared to the mRNA complementary strand. The ribosome shifts by a length of one codon in the 5' to 3' direction after peptide bond formation. Yes, this is what happens. You have got what we call the E site, the P site, and the A site. So the first amino acid that comes, the first tRNA that comes always goes in the P site, always in the P site. The rest of the amino acids will be the tRNAs carrying an amino acid will be coming in the A site. So once you form a bond between the one that is in the A site, the amino acid in the A site, and the one in the P site, what happens is that you are now going to shift this complex in this direction by one codon. One codon is also known as three nucleotides. So that is that. And codon of tRNA binds to RNA, not rRNA but mRNA. The sigma subunit of RNA polymerase detaches from the holoenzyme. So the sigma subunit of RNA detaches from the holoenzyme. That's not true. The sigma subunit is this unit subunit that was directing where replication is supposed to, transcription is supposed to start. It doesn't detach during elongation. The process is not over. Lactoperon is switched on when allolactose is bound to allolactose. Lactose is supposed to be bound to the repressor, as we said earlier, and then that means that lactoperon is turned on, meaning that the repressor is now away from the operator. That is what we call the on. Which one of the following statement about protein synthesis is correct? All the information in DNA codes for proteins. Again, this is not correct. The mRNA formed by transcription of a region of DNA only contains information for the protein to be synthesized. Both strands of DNA are transcribed. This is not true. Eukaryotic mature RNA is translated in the 3' to 5'. No. Okay. The eukaryotic mature RNA is translated. Translation is you are forming proteins. When you are forming proteins, you read the strand in the 5' to 3' direction. The first amino acid to be added is glycine. No, it's methionine. So the mRNA formed by transcription of a region of DNA only contains information for the protein to be synthesized. Let's say, for example, I want to make collagen. It's only for that protein. A base substitution mutation in a gene sometimes has no effect on the protein the gene codes for. Which of the following statements explains this? A base substitution mutation in a gene sometimes has no effect on the protein the gene codes for. So some codons specify more than one amino acid. Some amino acids are specified by more than one codon. So yes. So look at this question. A base substitution mutation sometimes has got no effect on the protein. That's for example, the strand that you add initially was 5'. You would say CCU 3'. And then after a mutation happens, you have 5' CCC 3'. Remember we said that the third position wobbles. So let's say the first here, the amino acid you are forming is A1. Even after mutation, you are still forming the same amino acid A1. And that is because some amino acids are specified by more than one codon. So the genetic code is redundant, it is degenerate. Identify the property of water that enables living organisms to cool their body surface temperature on a hot day. So on a hot day, it is a heat of vaporization and it is hidden. So we call it latent. That's the meaning of hidden. So it's the water has got a high latent heat of vaporization. What happens that by the time you lose water, it means that you've lost a lot of heat from your body. Heat capacity is important in maintaining a constant internal environment. Surface tension is important for moving of insects. Cohesion and adhesion altogether is going to be important for capillarity, moving water up a tube. The change of the state of water from gas to solid is called. So let's talk about this. This is solid. This is liquid. This is gas. So liquid to solid, what do you call that? Melting. Solid to gas, what do we call that? Sublimation. And then liquid to gas, that's evaporation. Gas to liquid, that's to condense, condensation. Evaporation is also known as vaporization. Solid to liquid, what is that? That's the melting now. Okay. Liquid to solid, that's freezing. This is melting. And then back like this, that is of course sublimation. Sister chromatids split during, so we are saying condensation because we're going from gas to solid. Sister chromatids split during, the chromatids, these guys, we call these sisters. So during anaphase, anaphase for away, they are going to be pulled away from each other. So that's anaphase. Longest phase of the cell cycle, already said that one is G1, which is part of interphase. All the following are different parts of eukaryotic chromosome, except a eukaryotic chromosome is like this. This guy is a chromatid. This guy is a chromatid. They are held together on a centromere. Okay. And it's actually on the centromere where I've got what we call the kinetochore. So chromatid is there in a eukaryotic chromosome, kinetochore, centromere. You also have DNA because for to have a chromatid is made up of DNA. But centrio, these are bodies that you'll be using to help in cell division. They're the ones that will be pulling chromosomes away via these spindle fibers. These are the centrioles. Proteins are said to be amphoteric because of the presence of both amino and carboxyl groups. So amphoteric means both hydrophilic and hydrophobic. Okay. And this, sorry, not hydrophilic and hydrophobic, but both acidic and basic. So because of that, because when you look at a protein, it has got this end is basic, this end is acidic, this is amino end, this is a carboxyl end. Amphoteric means acid and base at the same time. DNA replication occurs during the dash of the cell cycle. We say that's the S phase, which atomic orbital is dump, dumbbell shaped. It is the p orbital. It's like this. This is how a p orbital looks like. That's dump shaped. That's the dump bell shaped. The s orbital is oval. So it doesn't matter which p orbital, as long as a p orbital, whether 1p, 1 2p, x, 2py, 2pz. Select a statement that is true about plasma membrane. All living cells are surrounded by a plasma membrane. Is that true? Select a statement that is true. Not all living things. You have got some fungi, even some bacteria, they are living, but they don't have a plasma membrane. Cells receive chemical messages from the environment through the plasma membrane. Okay, so I'm gonna put a dot there. This is actually true. Now, this one, all living cells are surrounded by a plasma membrane. Okay, in this case, we're taking cells which are living. We can say yes, all of them, but to be specific, you have got some bacteria. Unless we say those are not living. Plasma membrane have protein channels through which nutrients for the cell pass. This is true. You have got these channels. Cholesterol is part of the plasma membrane. This is true. You have got proteins, carbohydrates, lipids. All the statements are correct. All right. The last question here. Which of the following statements is false about purine and pyrimidine bases? They are hydrophilic. So not most of them have got a lot of carbon hydrogen bonds, so that will be hydrophobic. Actually, they are hydrophilic indeed. This one is going to be true because of the bonds that like we are going to see them maybe something like this. Some of them have got nitrogens, and because of those nitrogens, they can interact with water, making them hydrophilic. Want something that's false. They are components of nucleic acid. That's true. Purines have two rings. Yeah, purines are double ringed. You have got one ring which is six membered, and then you have got one ring that is five membered. Pyrimidines have got only one ring which is six membered. Purines have one ring, so this is the opposite. They are heterocyclic. Yes, heterocyclic means that with a chain you have got different atoms. You don't have only carbons around. You have nitrogens, carbons, like that. That's a heterocyclic. So that is it with 2024 BIO 1400 exam.